is Morolake Thompson, or MK, and I attend the High School for Health Professions and Human Services. I'm working at the Albert Einstein College of Medicine with Luciano D'Adamio, and my co-mentor is Robert, who was one of the MD PhD students in the lab. The title of my project is Transgenic Mice Used to Study Alzheimer's Disease, or AD. This is basic information on Alzheimer's. I've been in the lab for about 2 weeks now, so I've just basic information. Alzheimer's is a fatal brain disease, and it's a form of dementia that causes nerve cell depletion, and it usually only affects people who are 65 years or older. And then you have the APP gene. The APP gene shown here provides the instructions for creating the amyloid beta precursor protein. So the enzymes, the beta secretase enzyme and the Y secretase enzyme, cut APP until it forms the amyloid beta precursor protein. The APP gene is located in the spinal cord and in the brain, which are the central nervous system. And Alzheimer's disease's connection to the APP gene. Alzheimer's is caused by genetic alterations that occur in the amyloid B peptide, or the A beta, in the production of the amyloid beta peptide, which are due to mutations in the APP gene. So basically, mutations here will obviously cause mutations here. There are about 20 mutations, but the most common mutation is in the amyloid precursor protein. This increases the production of the protein and causes plaques or clumps of the amyloid precursor protein to form, and this is what leads to dementia. We're currently doing a cell culture is created out of 293F T cells, which are stem cells, and we're growing ampicillin-resistant bacteria. Some of the mutations that occur in APP are the Dutch, the Flemish, the Arctic, the Swedish, the London, the Austrian, the Iowa, and the Italian mutations. They're basically named by the area where the mutation is, I guess, common or was discovered. And in this experiment, the wild type, which is the control group, is also used. But we're having a bit of a problem coming up with the wild type, because when we test for DNA, there seems to be no DNA. I'm not sure exactly what the problem is. Mini preps, which are basically they're the 20 microliter centrifuge tubes. Bacteria and ampicillin is placed into these tubes, and they're basically spun in the centrifuge until all of the materials, like the proteins and RNA, are removed except the DNA. The DNA and the restriction enzymes that cut the DNA at certain areas, and the buffers, which help make the enzymes more efficient, are added to a 96 well plate using a pipette, and they're put through an agarose gel electrophoresis, which uses ultra pure agarose, and this electrophoresis is ran through, and it ensures that the DNA is the correct size. And embryonic stem cells, which were previously grown, they have to be 90 to 95% confluent, which means that at the bottom of the dish or the plate, these cells have to stick to the bottom so that they can be used. And these cells will be transformed with the DNA that was provided through the ampicillin-resistant bacteria. The transformed cells, well, we haven't done this yet because we're still working on getting the rest of the materials. But what should be happening is that the transformed cell is injected into the early embryo or the blastocyst of a female mouse, and once female mouse gives birth, it will give birth to chimeras and like regular mice. And chimeras are mice that will have the mutated gene, and mice that will have the regular genetic information. Along the line, these mice are bred, and eventually there will only be a line of mice that have the mutated gene. The basic goal of the experiment is to use the information provided through the mice that have the mutated gene, to use this information to determine what causes mutations that occur in APP. And eventually, I guess this can help to find different cures for Alzheimer's, or to at least give us a better understanding of why it occurs and what occurs to cause Alzheimer's, and how can we try and stop it. These are my references, and I'd like to thank my mentor, Luciana D'Adamio, and Robert, who's been working closely with me in the lab, Zimara, Richard, Dr. Zat, and the Harlem Children's Society.
I wanted to know what exactly about Alzheimer's makes it fatal.
What?
I thought it was just
Well, Alzheimer, when it occurs, it sort of breaks down, it sort of like, let's say, eats at your neurons, so that your brain is not functioning very well. So once your brain isn't functioning, you're losing your memory. Other functions of your body won't be able to work correctly, because it's sort of, the mass of your brain decreases basically once your neurons decreasing, because they're not connecting. APP gene is mutated. It causes a mutation in the amyloid beta peptide. And once this mutation occurs, it causes clumps, or it causes this amyloid beta peptide to build up, and that builds up causes Alzheimer's. No more questions?